everyone. I'm Vincent Racaniello, and welcome to Session 2 of Week number 8 in Virology Part 1. This week we're talking about transcription, and in this video I want to explore the initiation of that process, how transcription actually begins. Here are the steps in the initiation process, which we'll go over in some detail in figures, but I just wanted to lay them out for you at the onset. First, there is what we call recognition of the core promoter, formation of a stable closed initiation complex, and that then becomes an open initiation complex, and then finally escape from the promoter. And under that, we'll talk about the regulation of Pol II by phosphorylation. We'll talk about promoter clearance, which simply means elongation, and the movement of the polymerase complex. So let's take a look at some of the elements of initiation now. The initiation takes place on a very specific part of the DNA template, which contains what we call transcriptional control elements. And here we're looking at the initiation of transcription mediated by polymerase II or Pol II, as we'll be doing for the remainder of this week. So here's our double-stranded DNA, and the initiation site for transcription or the synthesis of the mRNA is shown by this red arrow, and that would be +1, meaning it's base 1 on the mRNA. And the transcription occurs to the right, which means that the mRNA produced is by the polymerase as the polymerase moves to the right on the template. The sequence right around where initiation begins is called the initiator sequence. In fact, in this entire slide, what we're looking at are specific DNA sequences that play roles in the transcription process. Either sequences right at the site of initiation, like the initiator, or local sequences or distal sequences, as you can see from this slide. The actual core promoter, a sequence that you can take and have direct the synthesis of mRNA, is shown here. It consists of the initiator sequence and just a bit upstream the TATA sequence, and that's named after the fact that it has T's and A's and T's and A's in it. As you'll see, the TATA sequence is recognized by a very specific cellular protein. So the core promoter is about 20 to 35 base pairs in length, as you can see up on top here. The entire promoter, that is one which would be regulatable and responsive to stimulation or repression, and that's just called the promoter, also contains local regulatory sequences, which would be about 100 to 500 bases upstream from the initiation site. And that includes sequences that bind regulatory proteins that are shown here in green and brown and in orange. So again, the local regulatory sequences plus the core promoter would constitute the promoter region. And finally, we have distal regulatory sequences, enhancers and silencers, sequences that either stimulate or silence gene expression. And these can be quite far from the initiating site. They can be, as you can see here, 100 to 10,000 bases away. We'll talk about how those work later. So this whole region is called the transcriptional control region. And these are the sequences that regulate the initiation. Now, as I said earlier, transcription is largely regulated at the step of initiation. It's regulated by the interaction of various cell proteins with these regulatory sequences. And transcription needs to be regulated because in cells, of course, you don't want to have every mRNA being made at all times. You want to regulate them to make them when you need them. Sometimes you have to keep them off for long periods of time, or you have to turn them on quickly, or you have to regulate the amount of mRNAs that are made. And the same goes for viral infections. Not all mRNAs need to be made at one level throughout the infectious cycle. And so these regulatory elements, these control elements, allow cells and viruses to regulate the amount of mRNAs that can be made. So lots of work has gone into studying these to understand how gene expression, that is mRNA synthesis, can be regulated. So this is basically a summary of some of these promoter control elements so that you have it written down. Again, there are core or distal elements. Again, these are specific DNA sequences in the double-stranded template that regulate transcription. And one of them was the TATA box, which is a very specific sequence of TAs and TAs. And this binds a protein, which we'll talk about later, called TF2D. We'll be talking about a number of these proteins that are involved in transcription. TF stands for transcription factor. There's the initiator element, which specifies accurate starts, tells the polymerase exactly where to begin at the very 5' end of the mRNA. And there are also distal sites for regulation. These can be upstream, as we saw in the previous slide, but they can also be downstream, interestingly, of the mRNA start site. And then there are enhancers, position and orientation, independent DNA elements. These can be present and active in very specific tissues, or they can be everywhere. In other words, they're universal. So let's look at the initiation of transcription by Pol II. We start with a double-stranded DNA at the top. We're looking at the initiator sequence here, the core promoter, let's say. Here's the initiation site of RNA synthesis, the +1, the red arrow going to the right. So the first thing that happens is RNA polymerase II plus a series of initiation proteins bind to the core promoter. You see the Pol II is on the right here. These are all different initiation proteins. And this forms what's called a closed initiation complex. It's called closed because the DNA is still double-stranded. The next thing that has to happen is the DNA has to be unwound, and you can see that happening here. It has to be made 2 single strands. They're not broken, of course. They're melted sort of like DNA is when it's replicated at an origin of replication. In fact, one of these initiation proteins that comes in with the polymerase is a helicase that unwinds the DNA template. So now you have an open initiation complex. And then the polymerase begins to make short transcripts. It makes a number of short or abortive transcripts. And these are short and don't go very far because it's believed that the whole complex is constrained. It's attached still. The polymerase is still attached to all these initiation proteins, as you can see here. Can't move down the template, so it just churns out these short abortive transcripts. And then the contacts between the Pol II and the other initiation proteins have to be severed. And when that happens, then the Pol II can begin moving down the DNA template. It's called elongating at that point. And now you make a nascent mRNA. And of course, if the polymerase reaches the termination site, then the mRNA will be complete and will be released along with the polymerase. So those are the main steps in initiation. This happens, of course, in cells, whether or not they're infected with viruses. And this also happens on viral genes. Viruses use either parts or all of the cellular transcription apparatus to make their own mRNAs. Let's take a closer look at the closed initiation complex that is formed on a DNA template. Again, we're looking at a diagram of the DNA template with the TATA box and the initiator sequence that forms the core promoter. And you can see a number of these transcription factors. It's unfortunate that they're called factors because they are proteins, after all, but the name has stuck since the early days when they were first discovered. And there are many of these that come in along with RNA polymerase II to form this closed initiation complex on the core promoter. And you can see that this formation is causing a bend in the DNA. And in fact, the initiation of these, the binding of these proteins to the DNA is thought to cause this bend and form the closed initiation complex. The bend is thought to be part of the configuration of the DNA that would be needed to bring an enhancer element into place to stimulate transcription. But anyway, the bend is caused by the binding of these proteins to DNA. Now here you can see TATA binding protein, TBP, which is binding to the TATA sequence. It's a very important component of transcription initiation. And then one of the other transcription factors, TF2H, is actually the helicase that participates in the unwinding of the double-stranded DNA template to make it an open complex. So that's what the closed initiation complex looks like. We actually have the high-resolution X-ray structure of this. So here we have a structure of the TATA sequence bound to TBP. So it's not the structure of the whole complex, but rather of just TBP bound to the DNA. So you can see how it interacts. There are different ways that promoters, core promoters for Pol2 can be put together in viral genomes, and there are 4 different ones illustrated on this slide using adenovirus and SV40 as an example. Here for the adenovirus type 2 major late promoter, you have an initiator and a TATA box shortly upstream. Here's where the transcripts begin, a single initiation site. On the adenovirus type 2 promoter for 4A2, a different promoter, you have a similar initiator. It's yellow, showing there's homology in the sequence, but there is no TATA box. And there are 2 sites for initiation here, just to show you that you can have differences from one promoter to another. Now, turning to SV40, here's one example, the early promoter. SV40 has 2 patterns of mRNA synthesis, as we'll see later, in early and a late. And the early promoter has a TATA box and an initiation site, but no initiator sequence. The transcripts initiate in this region, nevertheless. And the SV40 late promoter is quite different in that it has 3 sites for initiation of mRNA synthesis, all characterized by the presence of an initiator sequence, which is different from that of adenovirus, and that's shown by the different colors. In fact, these 3 different initiator sequences are different from each other as well. And then none has a TATA box. So again, there can be quite a bit of variation in the core 2 promoter architecture. So I don't want you to think that the one image that I showed you initially is how all promoters are built. So far we've looked at the core promoter consisting of the initiator and the upstream TATA box sequence. Here on 3 different promoters are some of the upstream regulatory sequences that would, when added to the core promoter, make the promoter itself. And you can see on the adenovirus major late promoter, the initiator, the TATA box, and then we have 2 upstream sequences. And again, these are specific DNA sequences that bind very specific proteins that are involved in transcription. And these are given names like CP1 and USF. I happen to know that USF stands for upstream factor. These are on the SV40 genome, SP1 binding sites. Again, binding sites in the DNA for a transcription protein, a regulatory protein called SP1. You can see there are multiple binding sites for that. And on the adenovirus E2 early promoter, a slightly different arrangement, a TATA box right here, and upstream are sites for the binding of the E2F transcription factor and the ATF transcription factor. And again, this is about 100 base pairs upstream of the transcription start site. So again, not only the core promoter can be different, but the more proximal regulatory sequences that constitute the entire promoter can be different as well. Now, enhancers, as we mentioned earlier, are sequences that regulate transcription. They generally stimulate transcription, but these can be very far away from the actual promoter. And so here's a diagram of how we think this happens, and there is experimental evidence to support this. Here we have the RNA polymerase binding at the promoter along with all the transcription proteins, including the TATA binding protein. This, of course, is the initiation complex. The enhancer is a sequence which is hundreds to thousands of bases upstream, but nevertheless, it can interact with the initiation complex because we think the DNA bends. And remember, we showed how the initiation complex causes a bend in the DNA, and that may be in part to establish this configuration. So there are specific proteins that bind the enhancer sequence, which is shown here in orange. They're called enhancer binding proteins, and they in turn interact with the initiation complex. And the idea here is that the enhancers stimulate transcription, perhaps by regulating the stability, by increasing the stability of the initiation complex. So again, the enhancers enhance transcription, as the name would suggest, by facilitating these stable interactions.